Yeah, thank you all for having me. Thanks for the kind introduction. And with that, I will share my screen and we can go off to this presentation. All right, can I have confirmation that you can see the screen?
Yes, we can, Erik, yes.
Excellent, great. Yeah, thanks again for having me. So I realize that the time is not quite as long as I expected. So I probably have a few too many slides, but we can address questions at the end. Hopefully that's not a problem. This could probably be exposited over several hours, but the idea is to give a perspective on several ways of looking at these data structures, and also to clarify the definition of what is meant by a variation graph, that it's something actually very generic. And so that's something I hope to share today. So this is almost comical to begin this way, but for bioinformaticians, the kind of ultimate data structure you could imagine would be some universal tree of life. We can make different versions of these, of course, with the data we have, but we can't really get access to the actual object. That's what we're imagining when we do our work. And so in this thing, we have genomes that we're replicating over time. And when they have variants that arise through errors in replication, you get variation. And then that can give rise to diversity in genomes and eventually the diversity in the full tree of life that we see. And so that's all great, but we tend to forget it when we do analyses. We say, okay, well, it's simpler if I pick a reference genome that everything is pretty close to, and I'll use that to do my analysis. And the typical flow that I believe many of you would be familiar with is that you take the reference, you align sequences to it, and then you do variant calling process. That's basically just looking across the collection of alignments at every locus in that original FASTA sequence you have, and trying to find common variations between them and the reference. When those are common and they match our expectations of the way the genome of this individual works, or the pool, perhaps, collection of individuals, then we can call a variant, and we say this is a difference. And so every individual is described as a set of differences against the reference. And that's really powerful and very useful, but it breaks down when you try to do something that actually seems rather pedestrian, which is to update the reference to represent a diploid. So even that is not easy, because the way to do that would be to basically copy the reference sequence, put the edits into both, and then now you have two whole genome sequences representing your diploid individual. Even though you had this very nice succinct representation, not succinct necessarily, but compact. It's just the edits against the reference. So perhaps there's a better way, and there's a lot of things we could do with it. And why might you want to pursue such a thing? So this is sort of an initial exploration I made into this during the 1,000 Genomes project that, to me, completely clarified the need for this sort of approach. So here we have a locus on, it's probably chromosome 20, so then you can tell where it is. And there are 3 variants here that we detect with different, we differentially detect them with different variant calling methods. So there's the reference. That's pretty easy to see. All the variant calling methods, we have to detect that. Then there's a deletion of a lot of sequence, and you can kind of see that in the coverage plot on top of the screen here from IGV. And then inside of this deletion, like underneath it, so to speak, there's a small deletion of a single T. And so our variant callers are either detecting the alternate allele that's the big deletion, if they're an assembly-based method, or they're detecting the small one if they're alignment-based. And so you can actually homogenize this information by aligning all the reads at this locus to this graph using partial order alignment, and then projecting it back to the reference. And that's shown on the bottom, right? So just a proof of principle that we can actually see these variants in the alignments. And by the way, this is a mixture of different samples. It's kind of a pooled analysis just to validate it. So that's an interesting thing, because then maybe we can use a variation graph. So I'll define what that is in a second, but the kind of genome graph representing all the variation we know about as the reference itself. And then we wouldn't be biased to things that existed, that we knew about. And so that's important, because we're moving from a phase in which we have this kind of genomic approach where there is a reference genome. So I'm labeled here in R. I assume you can see my mouse. So this R, right, green. And so usually we align all the samples to this one genome, we find a difference. What's that? You can see the mouse. And so that's extending model is just aligning a new sequence in. But in the case of a pangenomic context, we might want something where we have like whole genomes that are assembled. We want all the mutual relationships. And then extending the model is equivalent to adding all the pairwise relationships between this sequence that we're adding and the rest of them. And the reference is just something we've picked arbitrarily. And so I'll argue that this approach can be achieved using a variation graph. And a variation graph is a graph. So it's built up of nodes that are words in the language modeled by the graph. In the case of DNA, they have DNA sequences attached, and they have identifiers, so we can find them and refer to them. And they tend to have arbitrary length, but they're at least not empty. And then edges are the topology of our model. They're describing how I can walk through the graph to emit sentences and phrases in the language of the model, or in the case of genomes, again, they're genome sequences. And because these nodes are actually bidirectional, they're representing reverse complement as well as a forward complement, these edges have the same property. They let us represent both the forward and reverse and invert between them. And the key piece that is important for the use of this as a reference system is embedding as walks things that we know about, genomes that we know about, perhaps that we built this graph from, perhaps a reference sequence that we use for coordinate bookkeeping, but something that we can use to anchor ourselves inside of the graph. Because as we build these kinds of structures and modify and extend them, we have to be able to provide mappings from that structure into things we know about. And so this idea is, they're called paths, but I guess in a graph theoretic sense, they're technically walks. They're just ordered series of nodes that we traverse in given genomes. And we can do any linear system in genomics can be projected into one of these. So a genome, a haplotype, alleles, and variants, they're just like collections of walks at the same locus in the graph, right? Annotations, intervals, those are like subsequences, and so those are walks as well. And so that, you know, in total, this model allows us to represent, to not just represent variation, but to losslessly encode a set of sequences and their mutual alignment, implying all the variation between them, and any kind of variation as well, any kind that we know of. Because the graph can have inversions, it can have cycles, it can have large-scale translocations, just as well as it represents SNPs and indels. And yeah, and so that a few ways to look at these things, right? We want to be able to see what we're doing. Seeing is believing in science as well. And so this thing I've been showing you is a kind of hierarchical layout of the graph using GraphViz. But we can also have layouts that are focused on the path space of the graph. This is the sequence 2 map, and it's designed to orient the structure of the layout to show you where the paths that are embedded in the graph go and understand the frequencies and the relationships. And it works well when the graphs are nicely ordered like this. We can break down more complex regions. And then there's also this an approach that's very scalable, very simplistic, which is to take the sequences of the nodes, concatenate them into one vector, basically, and describe every genome against them, every path in the data set against them, and remembering the topology in some way. So here, these bars that you see running across the x-axis are basically bit vectors. They're saying that this particular path or genome, or haplotype really, has presence at this node. And these encode a lot of the information we care about when we think about variation between genomes. And so bringing this together, there's another kind of layout I didn't describe, but some of you may be familiar with it. It's called, and the renderings will show are from Bandage, but it's a force-directed layout of the graph. And so putting this together in a case of an actual genome graph built from some HLA alternate alleles in DRB1 3123, we have this high-level two-dimensional view that shows the structure of the structural variants and so on. And that can be linearized if we sort it right into something that shows us how the paths relate to each other. And then if we were to zoom in on this, we can start to look at the actual bases. And one way to do that would be this linear view again, or perhaps something like the sequence 2 map. And so some of these ideas will come up in the talk. So this is also not a really novel idea to genomics. So the problem of relating multiple texts together affects people who do the research of collation, the relationship of different versions of text. And so in pursuit of making this easier, in the aughts, like more than 10 years ago now, there was research into building graphical models that were intended to compress different versions of the same sequence, the same text together, so that we could see the underlying structure in them. And so in this case, we have this poem that's about the Roman countryside, and you can see that we record where the different versions of the text go, just like the variation graph. And you can also look at it and see where there are variations and where there are things that remain constant through the many, many versions. So it's a way to show this all versus all relationship for 9 versions of this text that's very compact for the viewer. And in fact, it's called a variant graph. And the naming is coincidental. I wasn't aware of this until I was submitting my thesis, actually. And so this kind of arc of research has a pretty long trajectory. Pretty much since the ending of the 1,000 Genomes project, people have been talking about making a new version of the human reference that includes variation, so that we reduce our bias and when we use it for analysis, and we have a better perspective on new genomes. And so that this has been going on for some time. There are a few articles that come about it. My current postdoc advisor, Benedict Paten, was highlighted in one of them, and some of the early figures that I was making. This is a force-directed layout of a VG graph. It ended up in there. And it ended up on Hacker News. Some of you all may know. It's kind of like a Reddit or just a message board for discussion of technology. And the comments are often very snarky and very, you know, they assume after reading something like this that many things should be obvious. But this one was really interesting to me because it made me realize that what I was working on appeared to be an obvious thing to the average, you know, clever person who came and thought about it. They're basically saying like there's something we're missing, because clearly this is how you solve the problem, and like people didn't get so much smarter in 15 years. So why didn't they do this from the beginning? And you know, that's just uncomfortable. And so there was the response, though, which was to get together as a community and try to do it. So this was spearheaded by this paper, this is a consortium paper from 2018, and it lays out a foundation for, you know, what are the pieces that you need in order to re-implement basic bioinformatic analyses on top of this data structure. And not necessarily a graph, but a data structure that stores a pangenome. And so VG, the variation graph toolkit, is an attempt to make a reference implementation for many of these pieces, so we could begin to explore and understand what was useful and what was hard to do. And so we have pieces like the construction of graphs. So we have a bunch of individuals. We're going to put them into a graph to make a reference system, a pangenomic reference system. There's different ways to do that. I've been talking about this one at the bottom, which is a cyclic sequence graph where you label the paths that go through it. But you could also imagine even the underlying sequences make a pangenome. It's quite useful. Multiple sequence alignments tell you about their relationships, and they're equivalent to acyclic sequence graphs. And you can also build this is very nice because it's so scalable, a De Bruijn graph from those underlying sequences. That's something that you can build for very large collections of sequences. It still retains a lot of useful information. But I'll talk about this last one, as you can tell, based on my introduction. And as I mentioned before, it models both strands of DNA. So it's just a graph, but it's something that has intrinsic properties that make it look like DNA when you get up close. And so we can have inversions, and we can use assembly graphs as input. But we can also build it from BCF files. And so to get a sense of how that works, it's a little example for it. Build it basically by taking the reference, see a variant. That means you cut the reference around the variant and add in the alternate allele. And this works for both SNPs and insertions and deletions. And it's just kind of trivial. In fact, it shows that the VCF format is really a kind of graphical format itself. And in fact, a lot of tools that have worked in a pangenomic context, tools like Graph Typer, they don't actually instantiate the graph in the same way that I'm talking about. They really work on the VCF file, and that's enough. You know, that's enough to get a lot of information. So yeah, then we can index this in different ways. I won't get into the details of that because it gets quite into the weeds. But we have text indexes and indexes of the Text index would be a way to look up sequences in the pangenome, indexes of the set of haplotypes, indexes of the graph structure itself. And then also we can, so I'm having trouble seeing yeah, okay. So mapping new sequences into the graph is a kind of critical thing as well. So how might that work? I won't get into details of this either because it can get pretty weedy. But the gist of it is that we, as a community, have developed techniques that allow us to map reads against, to map long reads against complex graphs. So we don't have any limitation in terms of the scale of the thing we're mapping, in terms of the complexity of the graph we're working on. And this is just an example of one particular case. We mapped this 43 kb PacBio read into a graph made from different yeast strains. And the bits in red are showing where I found my mem hits, my initial seeding for the alignment. You can see that there's these telomeric ends that get hit, so it's kind of spurious. And the blue part is my best alignment. And the green one is my second best. And just to give a sense of how even though we're working in this complicated structure, we can still be very sure of our ability to relate things to it when we have a lot of information on long reads. Okay, so then variant calling is really a schematic how to think about it, but if you have short reads or fragmentary reads, not a whole genome assembly, and you want to map it into the graph and add things to it, you could do something that looks a lot like how we typically do variant calling. You do realignment, you'll find places that differ against the graph, giving rise to a set of variant sites that you can then, this is the difference from standard methods, you can then augment the graph, you can add the variant in. And you can also describe the variant calling results as a kind of sample graph or diploid graph, let's say. And this is something we've used for structural variant genotyping and various kinds of things. Sorry, my daughter's pretty upset. She just came home. Hopefully she'll calm down. Otherwise, I have to close the door. All right. So then, yeah, as I said, we can update the graph, right? These indexes we have allow us to query and retrieve bits and pieces of it. Visualization. I showed you 20 examples of that. And simulation, annotation. You can kind of see how these things work now. So just as a way to explore this a little bit. So our first foray into this was to use the variants from the 1,000 Genomes Project to understand whether having information about the variants would allow us to do better at basic bioinformatic tasks like read alignment. So we built this graph with 85 million variants in it, and it's directed and acyclic because it's a variation graph built from a DCF file. That's fine, that's what we had. And first, the question is, like, can we practically operate on these things? Like what kinds of times are required, and how much penalty do we pay by making a graph? So the indexing process we have, not very fast, but so it's very slow to index this one linear genome is their basis, right? It takes about a day, at least the version we had. And constructing the graph, of course, takes a little while. These all have gotten better, but it's not super great. And the thing is, if we add all the variants in, it doesn't get like exponentially worse. It gets a kind of linear factor worse relative to the number of sites we add in, the increase in size of the graph. And so we take about 2 days to do the indexing. And if we're mapping reads against it, we take 50% longer. And interestingly, if you start to remove the lower frequency variants, and these are specifically variants that are less useful for your average analysis, let's say, because they're less likely to appear in a new sample, then the run times start to approximate those for the linear reference. You're really just paying a small factor, small cost over that. And so that's a principle that I think suggests that these methods will eventually become quite performant and just a default, because although you can see how bad my indexing for the linear reference is, it takes a whole day. It should probably only take like 20 minutes or 5 minutes. If I could have a small factor over that to get something with all the variants up to 1% frequency, I'd say it's probably worth it. So that gives an idea. Anyway, so then to understand if this actually helps, we simulated reads from a diploid sample, and we simulated from the actual haplotypes of the sample. And crucially, we remembered whether they were matching the reference or had variation inside of them. And that matters for a point that I'll bring up in a second. So these are receiver operator characteristics for different mapping configurations. The green ones are VUG against the linear reference. The blue ones are VWA against the linear reference. And the red ones are against this particular graph. In this case, it's built with a full variant set. the dark ones are, the dark lines are for paired end reads, and the other ones are single-ended. So you can see that there's a sensitivity increase from paired ends. And there's some actually maybe loss in precision, in fact, as you get a little bit confused by the pairing. And unsatisfyingly, we don't do better by mapping it to the graph. Actually, there's a cost. So where does the cost come from? And it comes from reads that tend to match the reference. You're getting confused about where they go, because the addition of variation means that there's more sites that look similar to each other. And so you can see that this kind of loss is more increased here. But then when we look at reads that contain any variant at all, then even a single SNP, then we universally do better. But all the ROC curves are better for VG against the pangenome. And it's a very dramatic effect in the case of single-ended reads. But even in the case of paired end, we have this bonus. And so then maybe we don't need all the variants. Maybe that's hurting us, right? We're adding lots of ambiguity into the system. So we investigated cutting out alleles below 0.1% frequency, and then below 1%, and then finally below 10%. And you can actually see that at 10%, the loss is so minimal, and the benefit from this common variation we're finding in our sample in the set so high that we do better across the ROC curve with VG on the pangenome. And this goes to speak to the complexity of deciding what to put in the pangenome. It's a kind of prior model for what you expect to see in your sample. And in that sense, it has to be balanced against your needs of your analysis. It can't be a non-informative prior or a very ambiguous one. Both of those will hurt your performance. And so that's great, but it's based on simulation. And so for the interest of linking this back to real data and making sure that this wasn't some artifact of our process, we looked at heterozygous sites in the sample in a real data set and counted how many reads mapped each allele. In this case, there's two mapping runs have been done. One is split between red and green. It's VG against I think it's the 1%, 1/10 of 1% pangenome, and the ones in green are not in the VCF file from the health and genomes. Sorry, I'm not explaining this very well. So these are alleles. They're bins of alleles by length relative to the reference. 0 would be a SNP. You can see that things are pretty much 50%. As you increase, they'd be insertions, decrease, it'd be deletions. And what you see with in blue, this is BWA and freebase, you have a strong reference bias that is evidenced by the fact that you are missing a large fraction of your alternate allele observations relative to what you get in the reference at longer indel lengths. And you see that with the green line as well. And you don't see it with alleles in the graph. There's very little bias whatsoever. And so just to drive this home, it's been confirmed independently by a number of studies. The one I'm most familiar with uses a bovine variation graph. And they do a very similar kind of analysis. Here, the alleles in the graph are in orange, and you can see a similar kind of effect. The exact way that they're doing tabulation is different, but overall, the result is confirmatory. So this is effectively saying that when you put something in the graph, and you use the tool that's designed to treat every bit of the graph as the reference, you don't have bias to the thing in the graph, which is what you hoped happens. And so just to extend this further, so I said these are meant to represent any kind of variation, right? So they should be able to represent whole genome alignments in the graph and still work with it. So using the cactus whole genome aligner, we built this reference graph out of 7 strains. And so this pangenome has all the variation, all the sequences between these. If you look at the large scale, it's this kind of hairball with big loops coming off of it. And if we were to zoom in on the junction, we see these are different chromosomes here. This is one chromosome in different strains. This is another chromosome in different strains. And they have this region of homology that brings them together in the representation of the graph. So it's representing kind of structural variation. But then if we zoom in more closely, it's partially ordered. It's directed and acyclic, right? And so this is just to show that even though the graph at the large scale can look complicated, when you zoom in closely, it has a kind of manifold property that it looks partially ordered. And you can work with it easily because of that. And so we built this to validate the process of alignment using VG. And so the result is positive that if you were to build a graph missing one sample, and you take the long read data from that sample and map it back, you do better than if you map to the reference assembly, or rather the reference strain assembly in this case, from the same study. And so what you're seeing here is the identity of each read is a dot. Each read is represented by a dot. Its identity against the variation graph, its identity versus the graph built, the linear graph, so to speak, from this one strain. And the kind of region up here in the corner where it's darker is showing you that you're having reads that are mapping much better. Maybe they're mapping full length against the variation graph, and they're not mapping full length against the S288C reference. And there's some reads that just don't map at all. So this is variation that's shared with other strains, but not S288C, that allows us to map the reads. And this is, so there's some applications I'll get into now. This is a case where so in ancient DNA, you don't have the possibility of improving read length because you're limited by the molecules in the samples you get. And those can be quite short, maybe 50 base pairs. And they're degraded, so they have lots of errors in their ends, typically. And so this has made it very difficult to analyze these data and to work with them. And they're hard to align and detect variation, in particular indels. So this is an example we found from the alignment of an ancient Yamnaya sample that had about 20x coverage. So we found places where with VG we could detect SNP with much closer to 50-50 balance between the alleles, but it wasn't the case for BWA. And what we tended to see was that there would be another variant nearby. BWA align is pretty good if there's just one SNP and a few errors in the read, but once you have an indel, in this case, so these have been reprojected into these variation graphs to exposit them, but this is a collection of reads from BWA, right? And so the reads don't actually span the indel. Like, in fact, it would have soft clipped this one base. Whereas in the case of VG, we can map through the indel, and this indel happens to be in phase with this C, and so we detect this at the same frequency as the A, whereas that's not the case in BWA. And this is something you can see in the aggregate as well. And this is kind of recreating the sort of plot we saw earlier. Here we don't really know what the true heterozygotes are, so it's a little bit difficult to do. But we can ask, can take a set of potential heterozygous variants, different allele lengths, and ask, so how many of them do we detect with either BWA align or VG? And you can basically see that the ability to detect indels is very minimal in the case of BWA align, and also because the reads are so short, and it's not as affected in BWA in VG. And finally, this lets us do some pretty interesting things. So we can look for, say, clinically relevant alleles that are composed of indels in ancient samples. And so we take the CCR5 delta 32 allele, which confers resistance to pathogens, but also may have some, right, it's everything in balance, let's say, but it's known to confer resistance to pathogens. And we can look in ancient samples. So in this case, we look in the Yamnaya sample, and we actually just cannot detect a single copy of this allele, this locus. You just don't see it, right? But here we can see that the individual is probably heterozygous for this. And in other cases, they're lower coverage, you can detect a few, whereas you don't detect any in BWA. And this is just a fact of the way the alignment's done. So it's a study we want to do is to extend this to look at a lot of other clinically relevant alleles across the ancient DNA collections that are available. Okay, and so another application is this kind of, hopefully this isn't offensive to anyone, but neoclassical bacterial pangenomics. So that it's a reference to the idea so the genesis of pangenomics is in prokaryotic genomics, where you have genomes that contain, maybe have very little identity to each other, like 60 or 70%, but they're from the same species. And so these kind of species complexes can have open or closed pangenomes, in the sense that in an open pangenome, each individual might have a lot of relatively novel sequence inside of it and only a small core with genome that's shared with all the other individuals in that species. Whereas a closed one would be the other way. They all have pretty much the same genome. And so it's just an exposition of this idea. Here we take 10 E. coli strains, build a graph using Minia 3, and align the reads from that data from the same data set back to do a kind of coloring of the graph almost. And so it's a lot like a colored de Bruijn graph approach. So this is just showing a fragment of the sorted pangenome, which is about 10 meg long. And you can see that there's lots of parts that are shared between most of the samples with some variation embedded, and then larger variable regions that are just represented in a few of the samples and not others. And so in general, you have something like a third or a quarter of the data is in this bin that's only one sample, about the same in this one that's shared by all. So this is the core pangenome and the accessory pangenome, and the rest is in kind of intermediate state, right? And so it's another fun example. This is really just a test of the alignment process. So we built what we thought would be a very nasty kind of graph, and it's from viral genomes that have been isolated in freshwater lakes on Svalbard. And if you zoom in, they look like little viral genomes. So I've just built an assembly from them, and they don't make one giant component. They have lots of little pieces, which with variation inside, which is what you expect, right? And so I did this just to validate that you could map better if you map to the graph than to the contigs of the set. And so the thing that's interesting is the mapping rate when you map to the contigs with PWA, it's not very different than when you map with VG. It's the mean score, though, represents how much of the alignment you're getting. It's much higher in the case of VG. And you can see that with this shift in density in this plot, that VG's scores tend to be higher. And so it's probably the case that it's mapping through junctions, allowing us to in principle, get better quantification of those sequences in our metagenome. And okay, so with what time I have left, I will dig a bit into the problem of actually building pangenome graphs when we have whole genome alignments. So there's a kind of driving concept in my work right now is this idea that we're moving from a period in which it's been really hard to get good assemblies of genomes and reconstruct them into one in which those will be abundant. And that'll shift the problems we have from the one of reconstruction to one of relationship and understanding the relationship between many sequences. And so building these graphs is actually a way to achieve that goal, because they let us look at many to many relationships very compactly. So, right, how would we induce a pangenome graph, not from a VCF file, but just in general? So I think a very simple way is to take a set of alignments between the genomes. You can manifest that abstractly in something called an alignment graph. So here we have these genomes. They're written with these different letters that are representing different sequences and input. There's red, the blue, and the green. And when we have an alignment between two of them that says that they're the same, we put a gray bar. Now, if I then collapse all these gray edges into single sequences and walk back through the graph following the paths in the original input, I make a variation graph, in fact. And this is something that I've implemented in the sequish graph induction kernel. That it takes a path file as input instead of sequences, and it emits a variation graph in GFA format. And it took a lot of effort, but it's something that's incredibly scalable. And pretty much every aspect of its design is focused on running in parallel and running using external memory when possible, so that we can do things like build a variation graph from 25 human assemblies in 15 hours, which isn't great. Perhaps we'll still come down, but it's a start. We need to be able to, we need a scalable tool to do this particular step. It's been a thorn in the side of imperative genomics for some time. And so the thing about this that's really nice is that the inputs are very minimal. And the parameter space is small too. There's a set of alignments, and there's a set of FASTA sequences. And then there's filters, right? You can remove very, very short fragments from the alignments that might help locally to make things simpler. But you could also remove alignments that just as a whole are kind of short, and because those might be spurious, right? So here I used Minimap2 to take those same yeast strains I was showing before and align them and make this graph without any alignment length filter. And what you get is this kind of black hole, like there's an error sequence in there that's attracting everything, because there's this transitive closure that's pulling all of the stuff together. Maybe everything representing a particular repeat class is representing a small part of the graph with very high coverage. And that's hard to think about and hard to work with. And so you can remove the short alignments, simply removing alignments less than 10 kb, and the graph unfolds, basically, and starts to basically represent the chromosome structure of the pangenome. And so, yeah, moving on. So then looking into this can be confusing because we can't see where the paths of the genomes are. But an easy way to visualize them is this linear layout I was describing. And so here we have at the top the topology of the graph. The x-axis represents a sequence space. And I'm using these kind of bit vectors. They're multicolored to make them easier to look at, to represent the different sequences. And I can label these too, so it's clear what strain they're coming from. And you can learn some stuff very quickly from this. Like I'm representing this all over cell alignment. I can see that there is a rearrangement of several chromosomes in this one strain. Happens to be a very diverged Malaysian strain, and that's something we can immediately pick up from this, rather than looking at lots and lots of dot plots. And so I won't get much more into this, but this is a very nice visualization and also analysis framework to work with, because you can project things into this linear space and work on a matrix representation of your data, which is very, very powerful. So just some more notes about the complexity of actually building these kinds of graphs. So if I just induce the graph from alignments, then I can get very complex local structures. And so what happens is that the alignments look slightly different. They're normalized differently, depending on which way I come into them, the way the liner is operating. And that causes collapse, because it can look like the same base is mapped together, in these kind of complex networks, and they generate things like this. So here we have this kind of URC of this interrupted microsatellite. And it's being looped through over and over by these different sequences. And occasionally they jump out and they pick up an A, or they pick up a C or something, and they come back. And this is fine in the kind of abstract sense, but it's very hard to write algorithms that work on graphs that have this local structure. This would break a lot of the alignment algorithms we use. Things would get trapped there. It's not a good scene. So a solution, and I'll describe this more on the next slide with pictures, is basically just to run an MSA algorithm across the graph in a chunked way. And the chunking is important because it requires us to sort the graph. I'll try to get into that. But the gist of it is I can take this raw graph, smooth it, and in the process, I also have the opportunity to get a kind of consensus, which is interesting. And to zoom in and get some sense of how this works, basically the consensus here, these paths are showing us where I actually ran my multiple sequence alignment. And these lines are showing us the relationship between the three graphs. And so this collapsed thing in the middle gets unfolded a bit. And then I have a consensus sequence everywhere that are in the MSA. Right? And another way to look at this is that so this is a piece of a pipeline that I'm developing. So it's first to scaffold the pangenome with a very fast alignment algorithm called EDIT, which is based on MashMap2 and EDLib. Then induce the graph with sequich and sort this using a kind of multidimensional scaling, and finally run this local POA algorithm across the pieces of it to get a smooth consensus and a set of consensus paths. Okay, so if I have a few more minutes, I'll describe a case study where we apply a lot of these. Yeah?
Sorry, just a couple of more minutes so that you also have time to take some questions.
Absolutely.
Please.
Okay, I'll go quickly here. I think it's possible. I can do it in 2 minutes. So basically I have a chimeric phage population. It's been evolved for 25 passages, and we have nanopore sequencing from 10. They're chimeric because these wild phages were fragmented. We made their genomes are ligated, and then they were passaged by bootstrapping them in lambda phage coproteins. And so then it's very hard to analyze these with traditional methods, because when you make an assembly and you have heterogeneity in your input pool, then you get different kinds of results. And so my collaborator, Franklin Nobrega, basically found me and said, I can't assemble these and understand what's going on. And so we took a different approach. And basically we found that sometimes the nanopore reads are covering full phage genomes. So they're showing a range of the length frequency distribution of nanopore reads, where we have these unexpected spikes, and they're correlated with different passages, right? The first 10 go red to yellow, and the last ones, these are ones taken after 25 passages, are very diverged. They have these different colors. And I can then take these, correct them using the aggregate read data from each passage, and build a pangenome graph out of them. And this is a kind of schematic of it. It's the common structure. This is a visualization of it. won't get into. And if we render like the orientation of each of these sequences against the pangenome, we can see things like inversions that occur at some rate among the set. You can see where this is going from blue to black, right? And then finally, we can do something really interesting, which is you can take this input as a matrix, right? It's a binary matrix, we'll say, and we can directly use that to build a neighbor joining tree. And this is a neighbor joining tree, and it shows us something that is completely expected, which is that the red stuff, which is at the beginning of our process, lies toward the root. The very evolved strains are far out. And there's intermediate things that are happening that are not unexpected. This is just a different view of it. And so, yeah, and also I can make a PCA. And the PCA kind of hints at something you can see if I had more time to show you the other ones, which is that these early passages have representatives in all these different clades. You can see that these red dots are all over the place. And the implication of this is that we're basically selecting on standing variation that's been introduced in this initial chimeric pool production. And that's something we're exploring, basically. And this, well, it's just another way of looking at the relationships. If you're interested, look in the slides. It's about pairwise relationships between the genomes. So with that, I'd like to thank everyone for your attention and to thank a lot of people who've helped me pursue this over the years and are pursuing it in their own ways. And yeah, thank you all.